To cover this competency, that is applications of molecular technologies like polymerase chain reaction and recombinant DNA technology in the diagnosis and treatment of diseases with genetic basis. I have already made a video that has covered the applications of polymerase chain reaction in the diagnosis of diseases with genetic basis. In that video, I have explained the conventional polymerase chain reaction, though there are many variants of polymerase chain reaction. In this video, I'll be covering the applications of recombinant DNA technology in the treatment and diagnosis of diseases with genetic basis. In order to understand the applications, we must first understand the process of recombinant DNA technology itself. See, the basic strategy in molecular cloning is to insert a DNA fragment or a foreign DNA. It could be from human or some other source into a DNA molecule which is called as vector, that is capable of independent replication in host cell. And the insertion of the foreign DNA into a vector result in the formation of a recombinant molecule which is composed of DNA insert. This resultant recombinant DNA molecule is also called as chimeric DNA, DNA chimera, or recombinant DNA plasmid. Recombinant DNA technology is basically a process of joining DNA molecules from two different sources and then inserting them into a host organism to generate products for human use. So what is required in recombinant DNA technology? We require vector and a DNA or the gene of interest which we want to insert into the vector. A gene is transferred to a carrier molecule which is called as a vector. The most frequently used vector is the plasmid. Plasmid is a circular double-stranded DNA molecule found inside the bacteria. This PBR322 is the first genetically engineered plasmid and is very widely used in recombinant DNA technology. It has origin of replication. It has 12 unique restriction sites. Some of them are shown here, that these are the restriction sites. And it has two selectable markers, ampicillin and tetracycline resistant genes, that is ampicillin and tetracycline resistant genes. So cloning vectors should have an origin of replication, multiple cloning site and selectable markers. The role of these selectable markers will be discussed later. The first step in recombinant DNA technology is to isolate the desired DNA in its pure form, which should be free from other contaminations. However, in normal cell, the DNA exists within the cell membrane and is also present along with other macromolecules such as RNA, polysaccharides, protein, and lipids. We can use these enzymes to get the DNA in its pure form. To obtain the DNA from bacteria, bacterial cell wall can be broken by using lysozyme, and the plant cell wall can be broken by using the enzyme cellulase. To obtain the DNA from fungus, the fungal cell wall can be broken down by the use of the enzyme chitinase. In order to obtain the DNA from animal cell, one can use chemical, physical, mechanical disruption methods. Ribonuclease is used to remove any contaminating RNA. Protease is used to remove proteins such as histones that are associated with DNA. There are 5 steps in recombinant DNA technology. The first and the foremost is to obtain the DNA that we want to be cloned, that we want to clone. So in order to have the DNA, we need to isolate the DNA. The sample tissue is first homogenized or broken down uniformly. Cells are lysed by using various lysis methods and by using suitable detergents. Usually this lysis is done in the presence of DNA stabilizer, which protect the DNA from getting damaged. The protein and RNA is removed by the use of the enzyme protease and ribonuclease. Finally, the ethanol is added to precipitate the DNA in the form of thread-like structure. The precipitated DNA is then spooled out and transferred into DNA rehydration buffer. The breakthrough that made recombinant DNA technology possible was the discovery and characterization of restriction endonucleases or restriction enzyme. Restriction enzymes are produced by bacteria as a defense mechanism against phages. Enzymes act like scissors, cutting up the DNA of the phage, as you can see here in this slide, and thereby inactivating the phage DNA and thereby inactivating the phage. Restriction enzymes, they do not cut the bacterial DNA, as it contain an other enzyme called as methyl transferase. The function of the methyl transferase is to transfer the methyl group to the restriction sites present in the bacterial DNA. Once it is methylated, it cannot be recognized by restriction endonucleases. Importantly, restriction enzymes do not cut randomly. Rather, they cut at specific DNA target sequences, which is one of the key features that make them suitable for DNA manipulation. Any DNA molecule from viral to human contains restriction enzyme target sites purely by chance, and therefore may be cut into defined fragments of a size suitable for cloning. As I have already mentioned that restriction enzymes act as molecular scissors that cut DNA at specific locations. Restriction enzyme digestion involves the incubation of purified DNA with the restriction enzyme. In this case, we have taken the example of EcoR1 at the condition optimal for that specific enzyme. The condition is chosen such that the EcoR1 is functional. This action of restriction enzyme on the DNA is called as restriction enzyme digestion, and the specific location at which the digestion occurs is called as recognition site or restriction site. There are three classes of restriction endonucleases, namely type 1, type 2 and type 3. Among them, type 2 is most commonly used in recombinant DNA technology because type 2 restriction endonucleases recognize specific sites and cleave just at these sites. Moreover, type 2 restriction endonucleases do not require ATP for the hydrolytic cleavage of the DNA molecule. In this figure, the EcoR1 is the restriction enzyme which is used to cut this double-stranded DNA. It is to be noted that EcoR1 recognizes and cleaves the DNA after G in this strand and before G in the other strand. And the sequence which is recognized by EcoR1 is GAA-TTC, and the other strand has the complementary sequence. Similarly, the restriction enzyme EcoR1 cuts a circular DNA molecule bearing a single target sequence of the recognition site, resulting in a linear molecule with single-stranded sticky ends. We will discuss about this sticky ends later on. The principle is simply that if two different DNA molecules are cut with the same restriction enzyme, both will produce fragments with the same complementary sticky ends, making it possible for DNA chimeras or the recombinant DNA to form. Hence, if both vector DNA and donor DNA are cut with EcoR1, the sticky ends of the vector can bond to the sticky ends of the donor fragments when the two are mixed. And this is the second step of recombinant DNA technology. This sequence, which is the sequence recognized by the restriction endonuclease EcoR1. This sequence is present at 5 sites in the DNA of bacteriophage lambda. So, EcoR1 digests lambda DNA into 6 fragments, as you can see here, which ranges from 3.6 kilobase, 3.6 kilobase to 21.2 kilobase long. These fragments can be separated according to the size by gel electrophoresis. Gel electrophoresis is a method in which molecules are separated based on the rates of their migration in an electric field. The smaller molecules or the smaller fragments moving ahead of the larger as compared to the larger fragments. Fragments of interest can be extracted from the gel and can be used for further manipulation. However, the restriction endonuclease digestion of larger genomes yield quite different results. For example, the human genome is approximately 3 into 10 to the power 6 kilobase long, and is therefore expected to yield more than 500,000 EcoR1 fragments. Such larger number of fragments cannot be separated from one another. So agarose gel electrophoresis of EcoR1 digested human DNA yields a continuous smear rather than a discrete pattern of DNA fragments. Because it is impossible to isolate a single restriction fragments from such digests, restriction endonuclease digestion alone does not yield a source of homogeneous DNA suitable for further analysis. Quantities of such purified DNA fragments, however, can be obtained through molecular cloning. Both PCR and molecular cloning are the way to amplify the gene of interest. Most of the time, the two techniques complement each other. DNA cloned directly from sample is usually faithfully copied and fully functional, and therefore can easily be manipulated. Whereas polymerase chain reaction amplification may induce error. But it all depends upon what experiment one intends to perform. As I have already mentioned, that restriction endonucleases cleave specific sites in the DNA. The restriction sequences recognized by restriction enzymes are generally 4 to 8 base pairs long or 4 to 8 base pairs in length. These sequences are palindromic. Palindromic is a sequence that read the same on the two strands of the DNA when we read from 5' to 3' in one strand and 3' to 5' in the other strand. We know that restriction enzymes cleave specific sites in the DNA. Now I'll let you know the types of cleavages possible by use of restriction endonucleases. Some enzymes leave single-stranded overhangs or hanging regions at either 5' end or 3' end and are called as sticky ends, staggered cuts, or cohesive ends. These overhangs are complementary to each other. For example, in this first example of hint 3, digestion 5' to 3' AGCT sequence is complementary to the 3' 5' TCGA sequence of the other strand. However, this enzyme produces 3' protruding ends in the two strands of the DNA molecule that are complementary to each other. Another type of cleavage produces blunt ends or even cuts. Some enzymes are blunt cutters which cut straight down the middle of a target sequence and leave no overhang, as can be seen in this example. So restriction endonucleases, they scan, recognize and cut at the specific location. Often these blunt cutters or often these blunt ends are made sticky and by adding linkers or adapters. Linkers and adapters are short, artificially synthesized pieces of DNA which contain restriction enzyme site. I'll not go into the details of the linker and adapters. A multiple cloning site is also called as polylinker, is a short segment of DNA which contain many up to 20 restriction sites. This is the standard feature of an engineered plasmids. It should be noted that restriction sites within these multiple cloning site, that is the polylinker, are typically unique, occurring only once within the given plasmid. The purpose of this MCS in a plasmid is to allow a piece of DNA to be inserted into that region. In this figure, PUC18 vector is shown with 10 restriction site in the MSC, in the multiple cloning site. A multiple cloning site is found in a variety of vectors, including cloning vectors to increase the number of copies of a target DNA, and an expression vectors, which is also called as expression construct. It could be a virus or a plasmid. Its function is to express the protein in a cell. Expression vector should have all the properties of a cloning vector, that is the origin of replication, selectable marker gene, and multiple cloning site. In addition, it should also have a strong inducible promoter, transcription and translation initiation sequence, elements of transcription, purification tag, which is usually histidine tag, and fusion protein. Vectors are among the essential tool of gene cloning or gene expression. Specifically, a cloning vector is DNA taken from virus such as lambda-phage or the plasmid or cells of higher organism to be inserted with a foreign DNA fragment for cloning purposes. Major types of cloning vectors include plasmid, bacteriophage lambda, cosmid, yeast artificial chromosome, bacterial artificial chromosome, human artificial chromosome. For some studies involving analysis of genomic DNA, it is desirable to clone even larger fragments of DNA than are accommodated by bacteriophage lambda or the plasmid. Cosmid and yeast artificial chromosome vectors can be used for this purpose. So there is a specific reason for choosing a particular vector. It is important to note that the purified DNA of interest and the vector of interest are cut with the same restriction enzyme to produce the complementary sequences between the DNA and the vector. So this is the vector, this is the foreign DNA. Both are cut with the same restriction enzyme, that is EcoR1. The process of joining these two complementary sequences, that is this one and this one, using the enzyme called ligase is called ligation. The resulting DNA is recombinant DNA, and it is the third step in the recombinant DNA technology. In organism, DNA ligases join together fragments of newly synthesized DNA to form a seamless strand. DNA ligases in cloning do the same thing. If two pieces of DNA are having matching ends, then DNA ligase can join them together to make an unbroken molecule. As you can see in this figure, using ATP as a source of energy, ligase catalyzes a reaction in which the phosphate group sticking out of the 5' end of one strand is linked to the hydroxyl group sticking out the 3' end of the other. This reaction produces an intact sugar phosphate backbone. In this figure, two blunt ends are joined using DNA. Once the recombinant DNA has been formed, then it has to be introduced into the recipient host. This process is called as transformation, which is the fourth step of recombinant DNA technology. Normally, the bacterial cells do not accept foreign DNA molecule easily. Therefore, they are treated so as to make them competent to accept the new DNA. Bacterial cells can be made competent either by using the physical method, that is electroporation, or the chemical method, that is the use of calcium chloride. After it is in the host cell, that is the recombinant DNA molecule, once in the host cell, the plasmid vector is able to replicate because plasmids normally have a region of replication. However, now the donor DNA insert, as can be seen here, is part of the vector's DNA. The donor DNA is automatically replicated along with the vector. Each recombinant plasmid that enters a cell will form multiple copies of itself in that cell. Subsequently, many cycles of cell division will take place and the recombinant vectors will undergo more rounds of replication. The resulting colony of bacteria will contain billions of copies of the single donor DNA insert. This set of amplified copies of the single donor DNA fragment is called the DNA clone. As has been already mentioned that cloning vector should have three essential features. As can be seen here in this slide, it has region of replication, many unique restriction sites within this selectable marker, and at least two selectable markers. One is this one and the other one is this one. In this figure, BAM-H1 restriction site is cut open by using restriction enzyme BAM-H1, and the DNA of interest is also cut by using the same restriction enzyme, that is BAM-H1. The DNA of interest is being inserted at this place. Upon insertion of gene of interest at this site, the tetracycline resistance gene is no more functional, and this process is called as insertional inactivation. Now we will see how these selectable markers are helpful in selection of transformed cell. This is the master plate. As after transformation experiment, we will get three types of colony. Non-transformed one, that is without the vector. We will get the transformed colony but without the recombinant vector. It will have the vector, but the vector will be non-recombinant vector. And the third colony will be the transformed one with the recombinant vector. The majority will be non-transformed without the vector. The non-transformed colony will not grow on tetracycline as well as on tetracycline containing medium or the ampicillin containing medium. So this colony can be easily excluded. Whereas the colony which has transformed but the non-recombinant vector will grow both on tetracycline and the ampicillin containing medium, and this can also be easily excluded. However, this colony which has been transformed with the recombinant vector will not be able to grow on a medium containing tetracycline, but it will grow on the ampicillin containing medium. So this process of selection is called insertional inactivation. As we can very easily select these colonies, and this is the fifth step of recombinant DNA technology. This aspect is highly useful in differentiating which cells have been transformed or which ones have not taken up the recombinant DNA. The recombinant DNA that has been introduced into the recipient host will produce DNA clones if it's a cloning vector. But if it is an expression vector, then it will express the protein under optimal conditions. This protein is called as recombinant protein. Now we will discuss the applications of recombinant DNA technology. There is wide range of applications of recombinant DNA technology, but here we will discuss its role in the treatment and diagnosis of diseases with genetic basis. The pharmaceutical products synthesized through recombinant DNA technology have completely changed the human life. Such products include human insulin, human growth hormone, clotting factors, interferons, and vaccine. There are many more such products are produced by recombinant DNA technology. Synthetic human insulin was the first golden molecule of biotech industry and direct result of recombinant DNA technology. Currently, millions of diabetics worldwide use synthetic insulin to regulate their blood sugar. Synthetic insulin is made both in bacteria and yeast. In order to get bacteria to make insulin, number of factors need to be considered. Genes in eukaryotes have introns. Bacteria do not have introns in their genes, so they do not have the biochemical machinery to make to remove introns. Also, the eukaryotic proteins are processed after translation to produce mature insulin. So, in order to get the bacteria make insulin, first the DNA is synthesized based on the amino acid sequence of the two chains of insulin, that is A and B. And it was initially synthesized by Hakura et al. In 1977. The two synthesized DNA molecules were inserted separately into two PBR322 plasmid vectors. The genes are inserted by the side of beta galactosidase gene of the plasmid. Plasmid also has tetracycline resistant gene. The recombinant plasmid, these two recombinant plasmids are then separately transformed into E. coli host. Tetracycline is added to kill off any untransformed bacteria. The recombinant host produced insulin, which is fused with beta galactosidase. So it has produced beta galactosidase fused with insulin A and as well as with the insulin B chain. Then this beta galactosidase was detached from both the insulin chain A and B. The detachment of this insulin A and B chain was possible because of the addition of an extra codon, that is methionine, which was added at the end terminal of each gene of A and B chain. After detachment, A and B chains are joined in vitro to reconstitute the native insulin by sulfonating the peptide chains with sodium disulfonate and sodium sulfide. Depending upon, so by this method, the active insulin was produced. Depending upon the type of protein product required to be produced by recombinant DNA technology, complementary DNA, which is also known as cDNA, or synthetic DNA can be ligated to the expression vector, which can then be transformed into bacteria for the production of desired protein. cDNA or the complementary DNA is synthesized from mature messenger RNA by using the enzyme reverse transcriptase. Now I'll explain the gene therapy. Gene therapy is designed to introduce genetic material into host cells to compensate for abnormal genes or to make a beneficial protein. If a mutated gene causes a necessary protein to be faulty or missing, gene therapy may be able to introduce a normal copy of the gene to restore the function of the protein. The normal gene may be inserted into a non-specific location within the genome, which is called as gene addition. This is the most common approach. The abnormal gene can be replaced by a normal gene, which is called as gene replacement. An abnormal gene can be repaired, which returns the normal gene to its normal function. Gene therapy may involve germ cells, but germ cells gene therapy is considered unethical. Therefore, currently gene therapy exclusively involves somatic cells, which involves the introduction of genes into somatic cells of an affected individual. There are two ways for inserting and expressing the gene into a target cells. In vivo gene therapy involves injecting the vector into the bloodstream. The vector then must find the target tissue and deliver the therapeutic gene. Alternatively, cells may be removed from the patient and the genetic material inserted into them in vitro prior to transplanting the modified cells back into the patient, which is an ex vivo gene therapy strategy. The potential use of gene therapy is to cure genetic diseases. For example, in cystic fibrosis, the intact cystic fibrosis transmembrane conductance regulator gene is targeted to lung tissues. In Duchenne muscular dystrophy, an intact dystrophin gene is targeted specifically to muscle tissues. Similarly, F9 gene that code for the coagulation factor 10 is targeted to hepatocytes to cure hemophilia B. Some of the commonly used vector systems for gene delivery include physical methods, for example, the use of the liposome, receptor-mediated endocytosis, retroviruses, adenoviruses, adeno-associated viruses, adeno-associated viral vectors, and herpes simplex vector. These vectors, they differ in how well they transfer genes to the cell. They recognize cells, they recognize and are able to infect, and whether they alter the cell's DNA permanently or temporarily. The most commonly retroviral vectors are used for targeting therapeutic genes to mammalian cells, as they can deliver new gene by infecting the cells. The viruses are modified so that they cannot cause disease when used in people. The key viral genes, that is these genes GAP, POL and ENV, are replaced by the human gene or the therapeutic gene. The long terminal repeats, that is LTR, are retained as it is required for the integration of viral DNA into the host chromosome. The 5 sequence required to pack the viral RNA in viral protein codes are also retained. For the detailed information on how the viruses are modified, please refer to your textbook. Gene therapy was first tried in 1990 at the NIH in Maryland to treat 4-year-old patient with severe combined immunodeficiency disease, commonly called as SCID. SCID is caused by genetic defects that affects the function of T cells. Depending on the type of SCID, B cells and NK cells can also be affected. There are several forms of SCID. Here I am discussing the form of SCID caused by the deficiency of the enzyme adenosine deaminase, which is commonly abbreviated as ADA. You must have read about this enzyme, that is ADA, in DNA degradation. ADA deficiency results in accumulation of adenosine, deoxyadenosine, and deoxyadenosine triphosphate. All these are toxic to lymphocytes. Treatment involves isolation of ADA genes from normal healthy cells, followed by cloning in bacteria and its introduction into the retrovirus. The retrovirus is genetically disabled. The retroviruses are then used to introduce the gene into the lymphocytes of the patient in vitro. This is the T cells with disabled ADA gene isolated from the SCID patient. Retrovirus infect these T cells and transfer ADA gene to the cells. These cells are then grown in culture to ensure the ADA gene is active. Genetically altered cells are then re-implanted into the patient to produce ADA, that is adenosine deaminase enzyme. This is an example of an ex vivo gene therapy. In vivo gene transfer is necessary when cultured cells cannot be re-implanted in patients effectively. Example of in vivo gene therapy in patients with cystic fibrosis, a protein called cystic fibrosis transmembrane regulator, that is CFTR, is absent due to a gene defect. In the absence of this CFTR, chloride ions concentrate within the cell and it draws water from the surrounding. As you can see here, the normal CFTR channel allows the movement of chloride ions from inside of the cell to the outside, whereas mutant CFTR channel does not allow the movement of the chloride ions from inside to the outside, thus causing sticky mucus to build up on the outside of the cell. And because this causes the accumulation of sticky mucus in respiratory tract and lungs. Cystic fibrosis is treated by in vivo replacement of defective gene by the use of adenovirus vector. The normal human gene is cloned in the bacterium. The normal gene, that is the particular DNA sequence, is inserted into the harmless virus. The virus used here is the adeno-associated virus. Virus is then dripped into the lung through a thin tube inserted in the nose. Virus enters the lung cell nucleus, and in the nucleus the normal gene is inserted into the lung cell DNA by the virus. The cells are now with the normal genes producing the normal functional CFTR protein. Similarly, the gene therapy can be used in familial hypercholesterolemia, Gaucher's disease, hemophilia A, B, and Turner syndrome. Gene therapy may hold a cure for many genetic diseases, but since 1990, only a small number of patients with variety of disorders have been treated with gene therapy with varying degree of success. Like every technique, gene therapy also has some advantages and some disadvantages. Gene therapy has the potential to eliminate and prevent hereditary diseases like SCID or cystic fibrosis, and many more. If someone is born with genetic disease, gene therapy may give life. It might eradicate diseases from future generation. It might eradicate genetic diseases from future generation. There are many disadvantages of gene therapy. Gene therapy do not provide long-lasting therapy. Immune response to the transferred gene stimulate the potential risk to gene therapy. Viruses used in gene therapy may cause immune responses, inflammatory reactions in hosts. Disorders caused by multiple genes cannot be effectively treated by gene therapy. There are two methods used for gene delivery: a physical method, which consists of gene gun and microinjections, and the chemical methods, which consists of the detergent mixture and liquefaction. As recombinant DNA technology was being developed, scientists continued to devise applications for the diagnosis of human diseases. They used restriction enzymes to cut DNA of patients with hemoglobin disorders in fragments for analysis. When comparing the fragment patterns from patients with and without sickle cell anemia, scientists discovered that patterns differ, thus providing a DNA test for sickle cell anemia. The DNA-based diagnostic tests can identify structural variants such as point mutation as well as dysfunctional and absent genes. They are also used to identify a recessive disease gene in unaffected carriers or in healthy people to estimate disease risk. Prospective parents may be screened and, if identified as a carrier of severe disease, may be advised not to have child or to go for the prenatal diagnosis. DNA-based diagnostic tests can be used to diagnose variety of genetic disorders. Here I am giving you the example of sickle cell anemia. Sickle cell anemia is caused by point mutation or the single base substitution, in which normally the beta chain of hemoglobin at the 6th position, it normally contains the amino acid glutamic acid. However, in the individual suffering from sickle cell anemia, it is replaced by another amino acid, that is valine. This change in DNA will be seen as A replaced by T. This replacement of A by T abolishes one of the restriction sites of MST2, whereas other restriction sites for MST2 are preserved. So, analysis of DNA from, therefore the analysis of DNA pattern of normal individual or the individual or the heterozygous individual or the individual who is the carrier of the disease and the individual who is suffering from sickle cell anemia will yield three different patterns, as can be seen here in this slide. Normal individual will have only one band corresponding to 1.2 kilobase, whereas the heterozygous individual will have two bands, one corresponding to 1.2, the other is corresponding to 1.4. Whereas the individual who is suffering from the sickle cell anemia will have only one band corresponding to 1.4 kilobase. This is because the person who is suffering from sickle cell anemia has lost one of the restriction sites for MST2 and therefore he is having only the larger fragment of DNA. In this slide, some of the disorders for which the diagnosis has been aided by DNA analysis has been listed. You can go through this list. DNA aided diagnosis can be used for the diagnosis of various other gene abnormalities where a mutation may, rather than deleting the pre-existing cleavage sites, creates a new restriction site. With this, I have covered the applications of recombinant DNA technology in the treatment and diagnosis of diseases with genetic basis. Thank you for watching the video.